Hello all, this is Dr. Gopal Chandra Senadi, Research Assistant Professor from the Department of Chemistry, SRM Institute of Science and Technology. You can reach me through email chandruji@srmist.edu.in. Before moving on to the enzyme catalyzed reactions, first we should know what is a catalyst and its role in organic synthesis. In general, a catalyst is a substance that increases the rate of a reaction without itself being consumed or changed. A catalyst increases the rate of a reaction by decreasing the energy of activation. There are three ways a catalyst can decrease the energy of activation. The catalyst converts the reactant to a less stable or more reactive species. You can see it from a potential energy diagram through progress of reaction and free energy. The one which is the black color represents an uncatalyzed reaction, whereas the green color represents the catalyzed reaction. So, if you look, a catalyzed reaction converts the substrate to a less stable or more reactive species, then it can convert into product. Thereby, the energy of activation has been decreased from uncatalyzed reaction to a catalyzed reaction. In the second one, the catalyst stabilizes the transition state. So here the transition state can be stabilized by a catalyst and eventually it can decrease the energy of activation. In the third type, the catalyst changes the mechanism of the reaction. So if you look again, in the black one corresponds to uncatalyzed reaction, whereas in the green one it becomes a two-step mechanism, whereas in an uncatalyzed reaction it is a one-step mechanism. But if you look on to a catalyzed mechanism, the catalyst changes the reaction pathway. A one-step becomes a two-step mechanism, thereby decreasing the energy of activation. So overall, how a catalyst can provide a more favorable pathway for a reaction. So it can increase the susceptibility of an electrophile to a nucleophilic attack. It can increase the reactivity of a nucleophile. It can increase the leaving group propensity by converting it to a weaker base. It can increase the stability of a transition state. Let us see each case with a specific example for better understanding. So, nucleophilic catalysis. A nucleophilic catalyst increases the rate of a reaction by acting as a nucleophile to completely change the mechanism of the reaction. A nucleophilic catalyst forms an intermediate by forming a covalent bond with the reactant. Otherwise, it is also called as covalent catalysis. Let us look at with an example for the conversion of ethyl chloride to ethanol using iodide as a nucleophilic catalyst. And let's look upon how this iodide is rolling out as a catalyst and increases the rate of a reaction. So in general, the mechanism for an uncatalyzed reaction for the conversion of ethyl halide to ethanol, just a typical SN2 displacement to get the desired product. So what if the nucleophilic catalyst is present in this reaction? So let us see. So I minus is a nucleophilic catalyst which is also a better nucleophile than OH minus, will initiate the reaction by typical SN2 displacement to provide ethyl iodide as an reactive species. Next, we all know I minus is a better leaving group than Cl minus. So a weak nucleophile OH minus can attack the ethyl iodide, thereby expelling I minus to give the desired product. So initially a single step mechanism in uncatalyzed reaction was turned out into a two-step pathway. Thereby I minus is acting as a nucleophilic catalyst and eventually it decreases the energy of activation. So let us look at another example for nucleophilic catalysis, which involves the hydrolysis of an ester to corresponding acid using imidazole as a nucleophilic catalyst. So the first step is the attack of imidazole to an electrophilic carbonyl carbon to form a tetrahedral intermediate. then delocalization of this oxyanion to form an acyl imidazole intermediate. Now, which on reaction with further with water will give your desired product. So here the key point you should understand, imidazole is a better nucleophile than water. And acyl imidazole, which is formed during the course of a reaction, which can hydrolyzes more rapidly than an ester because imidazole is a better leaving group than the phenolate ion. The next example, what we are going to see is an acid catalyzed reaction. In general, we all know an acid catalyst increases the rate of a reaction by donating a proton. So let us look at the example hydrolysis of an ester in presence of a bronzed acid. So the overall sequence happens to have two slow steps. The first slow step is attack of water to an electrophilic carbon, and the second slow step is elimination of methoxide anion as a leaving group. So, in order to increase the rate of a reaction, an acid catalyst should influence the two slow steps which occur in this ester hydrolysis. Now let us see how an acid catalyzes the first slow step. So a catalyst must increase the rate of a slow step. So first, in the first slow step, the carbonyl oxygen gets protonated, thereby increasing the electrophilicity of this carbonyl carbon, and it will be more susceptible to nucleophilic addition even with a weak nucleophiles. If you look at this uncatalyzed reaction, so a weak nucleophile cannot attack that easily to this partially polarized electrophilic carbon, whereas when it's protonated by an acid, so it can be completely polarized, so the electrophilicity is more at this carbon, so that a weaker nucleophile like water can attack, thereby increases the first slow step. The catalyst increases the electrophilicity of the carbon, thus makes it more susceptible to the nucleophilic attack. Now let us look how an acid catalyzes the second slow step. If you look at the uncatalyzed second slow step, the leaving group would be a methoxide ion, which is a strong base and a poor leaving group. Here, the catalyst decreases the basicity of the leaving group by coordinating into it by converting it into a weaker base like methanol, thereby increases its propensity to lead. This is how an acid catalyzes the second slow step. Now, you would be clear that there are two slow steps that an acid can increase its rate of a reaction. In the first one, the acid coordinates to the carbonyl carbon, thereby increasing the electrophilicity of the carbon so that a nucleophile can easily attack. In the second slow step, the acid can coordinate to the leaving group, thereby decreasing its basicity and converting it into a better leaving group. Now we move on to specific acid and general acid catalysis. So far we were looking upon an acid catalyzed reaction. So further, how the acid catalyzation further classified as specific acid and general acid catalysis. Let us look upon what is a specific acid catalysis. The proton is transferred to the reactant before the slow step. In general acid catalysis, the proton is transferred to the reactant during the slow step. And as it is evident from the potential energy diagrams, in specific acid catalysis, the proton is transferred before the slow step. And in case of general acid catalysis, the proton is transferred during the slow step. Comparing specific acid catalysis with general acid catalysis by taking the hydrolysis of an ester as an example. Here, let us see how the two slow steps in ester hydrolysis takes place during specific acid catalyzed addition of water and general acid catalyzed addition of water, as well as specific acid catalyzed elimination of the leaving group and general acid catalyzed elimination of the leaving group. So first, in case of specific acid catalyzed addition of water, the proton has been transferred to the reactant before the slow step, which is nothing but the attack of nucleophile. In general acid catalyzed addition of water, the proton from the acid is being transferred to the reactant during the slow step, which means that during the addition of nucleophile. So in the second slow step, specific acid catalyzed elimination of the leaving group, the proton is being transferred to the reactant is nothing but to the OMe before the slow step. Whereas in general acid catalyzed reaction, the proton is being transferred to the reactant during the slow step. It means via the elimination of the leaving group. So in general, a specific acid catalyst must be a strong acid and a general acid catalyst can be a weaker acid. So in what cases the reaction goes by specific acid catalysis? In what case the reaction goes by general acid catalysis? So if your acid is stronger, then preferably the reaction goes by specific acid catalytic method, and if your acid is weaker acid, then the reaction goes by general acid catalysis. In case of specific acid catalyst, the protonation will occur before the slow step, whereas in case of general acid catalysis, the protonation will occur during the slow step process. So, which is very very important. So later on, we are going to look upon enzyme catalyzed reactions. All the enzyme catalyzed reactions will happen at the active site of the enzyme. So you will be having amino acids present at the active site of the enzyme, either it may be an acidic amino acids and basic amino acids. Those are moderate or weaker acids. So all your reactions will that occur at the active site of an enzyme, they go by general acid or general base catalysis. In order to know that, you should be well aware of these basics, so that with this knowledge we will move forward for enzyme catalyzed reactions in due course. Specific base catalysis. In general, a base catalyst increases the rate of a reaction by removing a proton. Specific base catalyzes dehydration of substrate hydrate. In specific base catalysis, the proton is removed from the reactant before the slow step. As you can see here, the hydroxide ion removing a proton from the substrate before the initiation of a slow step, thereby elimination of water to give the desired product. In general base catalysis, the proton is removed from the reactant during the slow step to afford the desired compound. How a base catalysis increases the rate of a reaction? Let us look in terms of transition state. You see in this particular structure, the transition state for elimination of OH minus from a negatively charged tetrahedral intermediate. The other one is a transition state for elimination of OH minus from a neutral tetrahedral intermediate. Okay. If you compare among these two, which transition is much more stable, it is clear the one which is on my left is more stable. The reason being, the transition state for elimination of an hydroxide ion from a negatively charged intermediate is more stable because a positive charge does not develop on oxygen atom. Metal ion catalysis. Metal ion catalysis can have three types. Type A metal ion makes the reaction center more susceptible to receiving electrons. So if you look here, the carbonyl oxygen gets coordinated to the metal, followed by increasing its electrophilicity of carbon, thereby a nucleophile can attack easily. In type B, a metal ion can make a leaving group into a weaker base by coordinating it with the leaving group. In type A and type B can also be achieved through general acid or specific acid catalysis by increasing the electrophilicity of the carbon by coordinating to this carbonyl oxygen, or by decreasing the basicity of leaving group by coordinating with the leaving group. Type C, metal ion can make water a better nucleophile. If you look here, the metal ion can bound to water to form a metal bound water, which will be in equilibrium with the metal bound hydroxide ion, which is a better nucleophile. Thereby it can attack weak electrophiles to initiate the reaction. So pK of metal bound water, if you look from this table, various metals from calcium 2 plus to beryllium 2 plus, as the pK value decreases. If you look for calcium, it's 12.7, and with for beryllium, which is 5.7, which is fairly acidic. So metal bond hydroxide ions are better nucleophiles than water. One example, metal-ion catalyzed decarboxylation for the substrate dimethyl oxaloacetate in presence of copper or aluminum as a catalyst. The reaction proceeds via decarboxylation of dimethyl oxaloacetate. The role of metal is to stabilize the negative charge developing on the oxygen via decarboxylation. So another example here is metal-ion catalyzed hydrolysis of an ester. So here the reagent used is zinc and water. The zinc will coordinate with water to form a zinc bound water, which will be in equilibrium with zinc bound hydroxide. That makes water better nucleophile to attack this carbonyl carbon of an ester to increase the rate of the first slow step to form a tetrahedral intermediate. So in the second slow step, the zinc will coordinate to the leaving group, thereby decreasing its basicity so that it can convert into zinc bound methoxide as a leaving group, which on further reaction with water to form a methanol, and zinc bound water is regenerated back for the next catalytic cycle. Eventually, the hydrolyzer product has been obtained. So, in a nutshell, metal bound hydroxide is a better nucleophile than water. The metal ion also decreases the basicity of living group. Before going on enzyme catalyzed reactions, let us look upon bird's eye view about enzymes. Enzymes are protein molecules in cells which works as a catalyst. Enzymes speed up chemical reaction in the body but do not get used up in the process. Almost all biochemical reactions in living things needs an enzyme. With an enzyme, chemical reactions go much faster than they would without the enzyme. This picture will give you a clear idea about how an enzyme is responsible for converting the substrate into product. So active site, which is very much important in an enzyme, where the substrate get used to fit and it will form an enzyme substrate complex. Further, that will give you a desired product and enzyme will be regenerated back for the next catalytic cycle. So we'll look in detail in the upcoming slides about what are all those terminologies. So before going in further, we should also able to understand the trivial names of an enzyme. The name of an enzyme usually ends in ase. It also identifies the reacting substance. For example, sucrase catalyzes the reaction of a sucrose. It also describes the function of the enzyme. For example, oxidases catalyze oxidation reaction. Could be a common name, particularly for digestion enzymes such as pepsin and trypsin. Let us look upon the systematic name. So, as we all know, IUPAC for common organic molecules and Hans Whitman nomenclature for heterocyclic compounds. Similarly, for enzymes, according to International Union of Biochemistry (IUB), an enzyme has two parts. The first part is the name of the substrate for the enzyme, and the second part is the type of reaction catalyzed by the enzyme. This part ends with suffix ase. For example, lactate dehydrogenase. So it is very clear. The first part is the name of the substrate for the enzyme. So here lactate is a substrate for the enzyme. And the second part is a type of a reaction catalyzed by the enzyme. This part ends with the suffix ase. So it is going to do dehydrogen and it ends with the ase dehydrogenase. So lactate dehydrogenase. This is how a systematic name for an enzyme is being generated. So what is easy number? Okay, enzymes are classified into six different groups according to the reaction that being catalyzed. The nomenclature was determined by the Enzyme Commission in the year 1961, with the latest update having occurred in 1992. Hence, all enzymes are assigned an EC number. EC is nothing but enzyme commission. Okay. The classification does not take into an account amino acid sequence, that is a homology, protein structure, or chemical mechanism. These EC numbers are four digit. Say for example, A dot B dot C dot D, where A is the class, B is the subclass, C is the sub of subclass, and D is the sub of subclass. The B and C digits describe the reaction, while the D digit is used to distinguish between different enzymes of the same function based on the actual substrate in the reaction. Let us look with an example. For alcohol, we all know NAD plus is nicotinamide adenine dinucleotide is an oxidoreductase. EC number, enzyme commission number is 1.1.1.1. No need to worry much about this enzyme commissions. These are very fundamental. If you can go through any textbooks, it can be clearly explained, which is self-explanatory. So let us move on further with the six classes the enzymes commission has been given. EC1, which is nothing but oxidoreductases. EC2 is a transferases. EC3 is a hydrolases. EC4 lyases. EC5 isomerases, and enzyme commission 6 is ligases. Let us see what all these types describes, and under each type what kind of enzyme catalyzer reactions can be performed in a simple IUB classification of enzymes. IUB is nothing but International Union of Biochemistry. Just we have seen a couple of slides earlier. So enzymes are classified according to the reactions they catalyze. So classes, six classes are mentioned here. Oxidoreductases, they do oxidation and reduction. Transferases, they do transfer of atom groups or atoms. Hydrolases, they do hydrolysis. Liases, they add atoms or remove atoms to or from a double bond. Isomerases, they rearrange atoms. Ligases, they use ATP to combine molecules. Let us see a little bit further in detail. So, the class EC1 oxidoreductases, the general reaction catalyst we all know, oxidation and reduction. So, the typical subclasses, it can do oxidases, reductases, and dehydrogenases. Oxidases is nothing but the function is oxidation, and reductases function is reduction, and dehydrogenases function is remove two hydrogens to form a double bond. One example under this class, use oxidation of ethanol to acetaldehyde using a particular dehydrogenase, alcohol dehydrogenase, and a coenzyme which is necessary for a reaction to occur at the active site of an enzyme. So this NAD plus, which is a coenzyme which will oxidize, which will get reduced to NADH plus, thereby oxidizing the substrate ethanol to acetaldehyde. The second type is a transferases. It is nothing but a transfer of functional groups. Further subclasses is transaminases and kinases. Transamination is nothing but transfer of amino group, and kinases is nothing but transfer of phosphate group. Let us look at an example for transaminases. This is a particular amino acid alanine and alpha ketoglutarate. If you use alanine transaminases, the amine group will be transferred from alanine to alpha glutarate. So you will end up with a pyruvate and a new amino acid, acidic amino acid, which is nothing but glutamate. So the third type is a hydrolysis. The general reaction catalyst hydrolysis reaction. Typical subclasses include peptidase, lipase, and amylase. And peptidase functions as hydrolysis of a peptide bond. Lipases functions as hydrolysis of ester bonds and lipids. I hope you all know what is lipids. Amylases hydrolyze the glycosidic linkages in amylosis. Okay, which is nothing but carbohydrates. The glycosidic linkages are predominantly found in carbohydrates. One particular example, you take a peptidase, is peptide, it is a hydrolysis of peptide. using water. So the polypeptide C terminal, we all know to cleave a C terminal, we need to have a carboxypeptidase A or carboxypeptidase B. The carboxypeptidase A will digest other than lysine or arginine, whereas carboxypeptidase B will cleave the C terminal uh, only lysine and arginine. Okay, so those things we'll look upon in the upcoming slides. So here you see it is selectively cleaving the C terminal to give your desired amino acid from the C terminal. Lysis, addition of a group to a double bond or removal of a group from a double bond without hydrolysis or oxidation. So they are typical subclasses: decarboxylases, dehydrases, and deaminases. Decarboxylases is nothing but removal of carbon dioxide. Dehydrases is removal of water, and deaminases is removal of ammonia. So one example for decarboxylases is pyruvate decarboxylase. So you use a pyruvate decarboxylase enzyme. Since you are going to decarboxylate the substrate pyruvate, so you will end up with acetaldehyde. So the fifth type is isomerases, the rearrangement of atom to form an isomers. They further isomerases converts this to trans, and epimerases convert D to L isomer. One typical example we can take is a malleate, which is a cis isomer. If you use a malleate isomerase, it will convert the cis isomer to a trans isomer fumarate. Finally, ligases, bonding of molecules using ATP, adenosine triphosphate. So synthetases and carboxylases. Synthetase is nothing but combined molecule. Carboxylase add CO2. Here we are going to add CO2 to pyruvate using ATP to stitch the molecule. Using pyruvate carboxylase is an enzyme. So you'll end up with oxaloacetate. ATP is converted into ADP, adenosine diphosphate, plus energy. Mechanism of enzyme catalyzed reactions. So a couple of slides earlier I have shown you a cartoon how an substrate fit into the active site of an enzyme and that delivers the product. Now let us look into in detail about the mechanism of enzyme catalysis reactions. So the proper fit of a substrate, yes, in an active site forms an enzyme substrate complex ES. E is nothing but enzyme and S is nothing but a substrate. So E plus S they form the ES. Okay, they will be in equilibrium. Within the ES complex, the reaction occurs to convert the substrate to product. So ES will further give the enzyme back with the product. The products which are no longer attracted to the active site are released. Overall, substrate is converted to product. So this is the overall sequence. Enzyme and substrate they are they will react to form an enzyme substrate complex which will be in equilibrium with the enzyme and substrate. Eventually, that will convert back to enzyme and deliver the product. So let us see the mechanism of an action. In the first step, this is an enzyme and this is the active site of an enzyme. Okay, look at carefully. This is an enzyme and the active site of an enzyme is like a normalized curve. Enzyme binds to the substrate molecule. This is a substrate molecule. Enzyme binds to the substrate molecule to form an enzyme substrate complex. So this is E, this is ES, and this forms a ES complex. Okay. In the second step, the formation of a transition state, a reaction without any transition state, the reaction will not give the product. Okay. Likewise, the ES will undergo form a transition state in the second step. The formation of transition state complex where the bound substance is neither product nor reactant. Okay, so the ES enzyme substrate will go to the transition state ES star. Okay, this is how the ES will form a ES star. So this is neither the substrate nor the product. Finally, it will deliver the ES star will deliver the product and enzyme recovered back. So there are two types of enzymes. So lock and key versus induced fit. So if you look at this enzyme is very specific and only suitable substrates can fit into the enzyme. Those models are called as lock and key model. And other one, induced fit, enzyme can change its shape at the active site according to the substrate, and it can fit to form an enzyme substrate complex. These are called as induced fit model. There are two types of mechanism. One is a lock and key, and other one is an induced fit model. So, what is a lock and key model? In the lock and key model of an enzyme action, the active site has a rigid shape. You can see the active site as a definite and rigid shape. Only substrate with the matching shape can fit into the active site. The substrate is a key that fits the lock. Okay, you cannot open a lock with a specific key. So this is an analogy for this lock and key model. Only a suitable substrate which has a similar size, similar shape of the active site can fit into it to form an enzyme substrate complex. On the other hand, the induced fit model of enzyme action, the active site is very flexible and they are not rigid. The shape of the enzyme, active site and substrate adjust to maximum the fit, which improves the catalysis. There is a greater range of substrate specificity. Okay, you look, the active site is of different shape in the enzyme, the substrate is of different shape, but when they form an enzyme substrate complex, they make a maximum fit by adjusting themselves. Okay, when there is a maximum fit between the enzyme and substrate, then the catalysis can happen in a greater extent to deliver the product. Now let us see the factors that contribute to the catalytic ability of the enzymes. Reacting groups are brought together at the active site in the proper orientation for reaction. The amino acid side chains that acts as a catalyst are positioned relative to the substrate precisely where they are needed. Amino acid side chains can stabilize the transition states and intermediates. Now, with this knowledge about catalysis, what we have seen from the start, like general acid catalysis, specific acid catalysis, general base catalysis, and specific base catalysis, and metal ion catalysis, and nucleophilic catalysis. Moving further, we have also seen a bird's eye view about enzyme classification and their nomenclatures. So with this knowledge, now we are going further, as discussed earlier, about the mechanism of hydrolysis of peptides, particularly carboxypeptidase A, which is an exopeptidase, by using the knowledge which we have gained in the catalysis. Okay, so I'm not going to discuss much about endopeptidases and exopeptidases, but I'm going to discuss the mechanism for an exopeptidase. The exopeptidase is nothing but the which cleaves the peptide at the C terminal. This is called as exopeptidase. So in that, carboxypeptidase is a reagent. So they may be either A or B. Carboxypeptidase A will cleave other than lysine or arginine. Carboxypeptidase B will cleave only lysine or arginine, which is present at the C terminal of a given peptide sequence. Okay. So the overall reaction, this is a polypeptide chain, and carboxypeptidase A is an enzyme and water. So it will hydrolyze, particularly the C terminal of the peptide bond is hydrolyzed and leaving the rest of the peptide sequence. Now let us look at the mechanism of carboxypeptidase A on cleavage of C terminal of a polypeptide sequence. So this is the active site of a carboxypeptidase A. The amino acids are positioned at the active site. So carboxypeptidase A is a metalloenzyme. Zinc 2 plus ion is a metalloenzyme in carboxypeptidase A, which is tightly bound to the amino acids which is present on the active site, like glutamate 72, histidine 196, and histidine 69. Also, it bonds to water to form a zinc bound water, thereby increasing the acidity of water to form a hydroxide ion. arginine 145 and tyrosine 248, they form a nice hydrogen bonding between the C terminal of the polypeptide sequence of the carboxyl group to stabilize the polypeptide chain inside the active site, and the side chain which is present on the C terminal phenylalanine, they nicely fit into the hydrophobic pocket of the carboxypeptidase A. That is why carboxypeptidase A cannot cleave when lysine or arginine are present on the C terminal of a polypeptide sequence. Now, arginine 127 and metalloenzyme zinc also coordinate to the carbonyl of the amide where the reaction is going to occur to cleave the C terminal, thereby increasing the electrophilicity of the carbonyl carbon by coordinating to the zinc as well as hydrogen bonding with arginine 127. Zinc will bound to water to form a zinc bound water. So here glutamate 270 acts as a general base catalysis by removing a proton from the zinc bound water to form a zinc bound hydroxide, which will undergo nucleophilic attack at the carbonyl carbon to form a tetrahedral intermediate. So the oxyanion formed also nicely stabilized by arginine 127 and zinc. Now delocalization back of the oxyanion will cleave the CN bond, and this NH minus will takes up a proton from glutamate 270 by acting as a general acid catalysis. In the next step, so once the peptide bond is cleaved, internal proton transfer to form NH3 plus and COO minus. Now, due to electrostatic repulsion between this carboxyl ion and the glutamate 270 COO minus, the formed or the cleaved peptide product will be expelled out of the active site of the enzyme. The amino acid has been cleaved off from the polypeptide. So I once again I repeat the importance of amino acids which are present on the active site of an enzyme. So once the polypeptide fit into the active site of an enzyme, so the amino acids which are present on the peripherals of active site, say for example arginine 127, which nicely coordinate with the carbonyl oxygen of the amide, which where the reaction is going to occur, thereby increasing the electrophilicity of the carbon. Likewise, the metalloenzyme zinc ion also nicely coordinating with the carbonyl oxygen of the carboxylamide, thereby increasing the electrophilicity of this carbon. Now, the zinc bound water increases the acidity of this water, thereby glutamate 270 acts as a general base catalysis to remove a proton from the water. Now the OH minus, which is nothing but zinc bound hydroxide, will initiate the nucleophilic attack. Likewise, the role of arginine 145 and tyrosine 248, they nicely stabilize the carboxyl group at the C terminal of the polypeptide sequence, forming a hydrogen bond. Now the side chain which is present on the C terminal phenylalanine nicely fit into the hydrophobic pocket. Now I would like to remind again, that is why carboxypeptidase A is very selective. It will not cleave when you have a lysine or arginine at the C terminal of a polypeptide sequence. I hope you understand the mechanism clearly, and I believe that we have started with catalysis and types of catalysis. In that, we have discussed about what is the key difference between general acid catalysis and general base catalysis, specific acid catalysis, specific base catalysis. We also looked upon metal ion catalysis and nucleophilic catalysis. And in metal ion catalysis, we have further seen how metal ion can increase the rate of a reaction by increasing the slow step. In the first way, the carbonyl oxygen gets coordinated with the metal ion, thereby increasing the electrophilicity of the carbon. In the second, it can increase the rate of a reaction by coordinating with the leaving group, thereby decreasing the basicity of the leaving group. And in the third, the metal ion can coordinate to water to form a metal bound water, the metal bound water increases the acidity of water, thereby it will quickly deprotonate to form as metal bound hydroxide, which will be in equilibrium with metal bound water, so that the metal bound hydroxide can easily attack the electrophile, even a weak electrophile. So based on this knowledge, we further moved on to enzymes, and we have seen how an enzyme will be useful to convert the substrate into product by fitting into the active site to form an enzyme substrate complex. Then we also looked upon the types of classifications, enzyme classification. There are of six types enzyme classification: one, two, three, four, five, six, and we have seen in detail each with an example. So I believe this 40 minutes of lecture would be very useful for you guys to understand the types of catalysis and further and how it is useful for the enzyme catalysis. Okay, the most important thing, how to distinguish between a specific acid and general acid catalysis is, if you use a strong acid, then it goes by general specific acid catalysis. It means before the slow step, the proton will be transferred or the proton will be removed from the substrate. And in case of general acid or general base catalysis, the proton will be transferred or removed during the slow step. Okay, when it comes to enzyme catalyzed reactions, so the acids what present on the active site of an enzyme or weak acids or weak bases. In general, weak acids or weak bases will facilitate general acid or general base catalysis. So we have ended up with by looking upon the mechanism of carboxypeptidase A by using glutamate 270 acting as a general acid or general base catalysis, and zinc as a metalloenzyme. It will bound to water to form a metal bound water. And I hope you all enjoyed this session. Thank you all. If you have any clarifications or doubt, definitely at my initial slide I have shown my email ID, so you can write me an email to just clarify your doubts. Thank you all. Stay safe. Thank you. Bye bye.